Open Box Science for the opportunity to present today. Let's see if I can advance my slide. Here we are. Today, I'm excited to share work that was recently published in Nature Immunology, featuring contributions from a very dynamic and highly collaborative team. And this project really seeks to understand how diverse microbial exposures, largely in the periphery, can influence memory T cell surveillance of the brain and modify the outcomes of a model neurological disease, in this case, a seizure. So to begin, I am a basic immunologist. I utilize mouse models to better understand aspects of human immunology. But I think we can all appreciate that there are ways in which the utilization of conventional laboratory mice may fail to recapitulate aspects of human immunology. And in thinking about these aspects pretty broadly, or these broad domains, I think we can appreciate that we often use inbred mice, so there may be a lack of genetic diversity. There may be a lack of natural environments or opportunities to exhibit exploratory behaviors, a lack of intra and interspecies interactions. But the domain that I'm going to be focusing on today is that of microbial exposures. Sort of what can be achieved beyond what is imparted in sort of conventional mouse housing facilities. And why is this a particularly important question to think about? Well, at least as early as 2016, it was appreciated that if you look at the peripheral immune profile of an SPF mouse, a specific pathogen-free mouse, that it is going to more closely resemble infant cord blood than adult blood. And one reason or one explanation behind this could be the fact that in humans from birth to adulthood, there are going to be multiple microbial exposures that are going to drive the maturation and development of our immune system. And so several groups have had very vested interests in understanding how we can kind of bring up the microbial exposure histories of mice to maybe better recapitulate what we see in humans. And so several approaches have been proposed. One idea would be to just study wild mice. This, of course, is very difficult. These mice may harbor pathogens that we don't want to bring into laboratory environments, and may also be very difficult to power. But some other solutions have been proposed that may be a bit more tractable. Here, it's possible to resource pet store mice that are going to harbor a natural microbial burden that could be transmitted via natural transmission routes to SPF mice to generate a group of co-housed mice. Additionally, another idea would be to sequentially infect mice. So you could effectively keep a group of mice as staying SPF and compare them to mice that are sequentially infected with known spacing and known pathogens that have known clearance dynamics, and then study these comparative specific pathogen experienced mice for their immune profiles. Now, these ideas have been around for a while, and I've tried to highlight in this lower left-hand corner several groups that have done a really great job of describing what happens immunologically after co-housing or sequential infection. And one of the most striking findings, at least in my opinion, has been the increase, both numerically, proportionally, and spatially, this greater distribution of memory T cells that you see, at least in mouse hosts. Now, we know that there are several fates that memory T cells can take after kind of transitioning from a previously naive state following antigen exposure. One route would be to become a circulating memory T cell that is constantly on the move, that is going to express a series of surface markers that will allow them to constantly surveil the blood, lymphoid organs, lymphatics, and non-lymphoid tissues. But we also know that there are some memory T cells that adopt a different way of life, that become tissue resident memory T cells and sort of stay in one tissue long term. And that these cells are going to express a series of surface markers that either directly characterize or contribute to the ability of these cells to be retained within tissues long term. Now, in the center image of this mouse, what I've tried to do is highlight tissues that have already been profiled by these previous papers. And you'll probably note sort of classic lymphoid organs like the spleen, lymph nodes, as well as a lot of barrier tissues like the respiratory tract, the gastrointestinal tract, reproductive tract, and integumentary tract like the skin. But of course, as an immunologist, I've appreciated that there's one organ that's always missing, and that is the brain. And so in these studies, we really don't understand how microbial normalization can influence brain immunity, and in particular, memory T cell surveillance of the brain. And in thinking about this question more broadly, how this microbial normalization or changes in immune composition could change aspects of brain health and disease. And so when thinking about why this organ has been maybe ignored for so long, I think there's always sort of these historical conventions of immune privilege. But as Tomomi mentioned in her talk, we have known that memory T cells can be identified in various compartments of the human brain during health and disease. And so here I'm just highlighting the fact that these cells have been identified in both like the parenchymal tissue, the white matter and gray matter of the brain of humans, as well as the cerebrospinal fluid. And actually in cerebrospinal fluid, T cells are the most abundant cell type. But I think what is always a barrier in studying human tissue is we don't necessarily know the originating events that got those cells there. And so origin has remained somewhat elusive. But work that's been led, I think, by the labs of David Gate and Pamela Rosado has really, I think, revealed via TCR sequencing and tetramer staining that at least a subset of these memory T cells exhibit specificities for really ubiquitous peripheral pathogens, such as influenza virus, cytomegalovirus, and Epstein-Barr virus. Bringing this back to mice, what we also know in thinking about generating T cells in the brain, such as those that are TRM, is that peripheral infections can drive this memory T cell generation, including viral infections, bacterial infections, including segmented filamentous bacteria that Tomomi mentioned earlier, as well as parasitic species. But I think we can simultaneously appreciate that single infections on their own are very informative, but may be insufficient to capture the physiologic and diverse pathogen exposures that humans are going to have across their lifespan. And so the question that we were trying to address with this study is if we could generate mice with polymicrobial exposure histories and study facets of brain immunity and disease. And so to do this, we first reached out to the lab of Thomas Griffith at the University of Minnesota. They have a model system wherein they are able to resource pet store mice derived from the local Minneapolis-St. Paul area, and they are able to co-house them with SPF mice for 60 days. And this will allow the generation of a co-housed group of mice that are going to have increased microbial exposure histories. In parallel, closer to home here at the University of Iowa, we were able to collaborate with the lab of Vladimir Badavinek, wherein we were able to utilize a sequential infection model wherein mice were infected 5 days apart with 5 highly tractable peripheral infection models. These include PR8 influenza, murine cytomegalovirus, vaccinia virus, attenuated Listeria monocytogenes, and LCMV strain Armstrong. And similarly, we analyzed these mice at day 60 or post that time point. Additionally, we were able to use IV exclusion by injecting mice with an anti-CD45 antibody prior to tissue harvest in the context of these experiments at the University of Iowa to ensure that the cells that we were looking at were indeed not in the brain vasculature at time of harvest, but in the brain tissue proper. So first, before we had any sort of idea that T cells would be the focus of this paper, we wanted to first assess broadly what changed immunologically following these microbial normalization regimens. So the data that I'm showing here is concatenated and downsampled flow cytometric data. And this is going to be derived from SPF and co-housed mice. Here we're pregating on brain immune cells that we're defining as CD45 intermediate to high via a UMAP projection. What I hope you can appreciate is that, as expected, the most abundant cell is going to be microglia in both of these hosts. These cells have been there since development. But what we do start to appreciate in our co-housed mice is that we're starting to see a proportional increase in peripherally derived myeloid cells, as well as T cells following the induction of this microbial exposure. And we're able to show here on the right, across many mice, what that overall numeric fold change was between co-housed and SPF mice. So then we wanted to ask, is this conserved in a different comparative model system of polymicrobial exposure? And so I'm setting up this slide the exact same way. The only discrepancy here is that I've added that IV exclusion. And if we look at our representation of immune cells, we may not see necessarily an increase in our myeloid population, but we do see a conserved enrichment of T cells in the brains of these microbially experienced mice. And this numeric fold change summary for specific pathogen experienced mice is shown here on the right. So what we were effectively seeing is at two different institutions, two different sets of hands with many different sets of microbes, was a conserved phenomenon of T cell infiltration and sort of permanence within the brain. And so we then wanted to follow up on this particular immune cell subset and ask, what is the identity of these enriched T cells? So the first discrepancy that we wanted to determine is whether these cells were naive or were they memory. And memory is really a definition that involves time and antigen experienced, being antigen experienced. And so one way we can determine whether cells are antigen experienced is via their differential expression of markers like CD11A and or CD44. And so if we look at brain CD8 T cells that were derived from SPF and co-housed mice, what I hope you can appreciate is that we're seeing an enriched population of antigen-experienced T cells in our co-housed mice. When we then look at the overall cell numbers, sort of compiling both that enriched T cell population in general and the fact that most of these tend to be antigen experienced, we start to see a very striking numeric increase between SPF and co-housed and SPF and specific pathogen experienced mice in terms of how many memory T cells are existing within this brain tissue. We also did this for our CD4 populations, here adding CD44 just to get a little bit of better resolution. And what we are similarly seeing in two polymicrobial exposure regimens is that co-housed and specific pathogen experienced mice are going to have these massive increases in overall T cell presence in the brain. So then we know that these cells are there. We then wanted to tackle the question of whether these antigen-experienced T cells are functional, as we know that many memory populations tend to have a more robust ability to produce cytokine following restimulation. And so to do this, we were able to look at our brain tissue and compare a no stimulation condition to a broad PMA ionomycin-based stimulation assay to assess the ability of these cells to produce cytokine. And what we had previously done is sort of look at at least among our CD4s, how they were skewing in terms of Th1, Th2, Th17. And what we saw was a really strong sort of type 1 profile. And so we selected these particular cytokines to stain for interferon gamma, TNF, classic type 1 cytokines. And what we observed is that if we looked at our antigen experienced CD8s and CD4s, there was this profound ability following 4 to 5 hours of PMA ionomycin stimulation to produce cytokine. This was true across co-housed and specific pathogen experienced mice among our CD8 T cells. And this was also true if we looked at our CD4 populations as well. So then we wanted to ask the question, these cells are enriched numerically, they are functional, are they actually resident within the brain? And so in some of the supplemental figures for both co-housed and specific pathogen experienced mice, we did stain for classic markers of tissue resident memory. But for a lot of TRM-based immunologists, The real test of whether a cell is resident is the fact that it doesn't move. And the test or assay that you can do to confirm that these cells are not moving is parabiosis. And so to do this, what we did is we took infection-matched mice that were allelically disparate at the Thy1 locus. So we had a Thy1.1 parabiont on the left, a Thy1.2 parabiont on the right. These mice were then surgically joined via parabiosis, such that they will form anastomoses, where blood will flow and form a shared circulatory system. And then 3 weeks later, we analyzed our tissue once we were able to confirm equilibration between the two hosts. What I'm showing here is data from the IV negative brain. We are looking at antigen experienced CD8s and CD4s. If we first look at the brain of a Thy1.1 parabiont in their CD8 compartment, we can see that the vast majority of T cells are going to be of self-origin. They are Thy1.1 positive, but it's not 100%. There are some cells that are coming from the partner mouse that are inside of the brain. In parallel, if we look at our Thy1.2 parabiont, we see a similar outcome. Most of these cells are of self-origin, but not all. And so what I think this summary data really shows us is that many of these cells are going to be resident in nature. But despite these sort of ideas of immune privilege, I do think that there are circulating memory T cells that are maybe able to dip in and out of the brain occasionally and circulate through this tissue. Then we wanted to kind of confirm that what I just said was true, that these cells that are of self-origin look like resident cells, that cells that are of partner origin maybe look like circulating memory T cells that are just coming in and out. And so to do this, we were able to stain these differentially originating cells with markers of residency, such as CD69, 49A, CXCR3, and CXCR6. And overall, what we were able to determine is that cells that were of self-origin were going to exhibit greater expression of these residency markers, suggesting to us that this sort of commitment to resident memory phenotype, probably that window sort of closes sometime after the resolution of infection, but that circulating memory T cells can continue to survey this tissue. And what I'm showing here below is just the summary data of these self and partner-derived cells for CD8 and CD4 T cells in terms of what that expression profile of these TRM markers looks like. So next, we appreciated that our previous studies had sort of treated the brain as sort of one homogeneous organ, but we know that there are distinct niches that look very different within this tissue. And so we devised a strategy wherein we were able to assess the parenchyma, cerebrospinal fluid, and choroid plexus as 3 sort of representative distinct regions within the brain to see if the expression of residency-associated markers would actually vary based on which area you might sample. And so first, we're showing flow cytometric data here, looking at our CD8 populations between the parenchyma, CSF, and choroid plexus. And what I hope you can appreciate is that the expression of a canonical TRM marker, CD69, does vary according to region, where parenchyma has the greatest frequency of positive cells. The summary data is shown here on the right across many mice, where again, we're seeing for CD8s and CD4s that this greatest proportion is within the parenchyma. But of course, we needed to validate this with parabiosis. And so we performed parabiosis and isolated these distinct brain regions and similarly confirmed that the parenchyma is going to harbor the greatest frequency of self-derived cells. And perhaps the CSF and choroid plexus may be slightly more permissive to circulating memory T cells coming in and out. We also did some immunohistochemistry to discern some regions of interest where these enriched T cells may be populating. So here we're looking at SPF, specific pathogen experienced, and we're bringing back our co-housed mice here. And what I hope you can appreciate by looking at white matter structures, which are going to be very lipid-rich structures in the brain, is that we see this increased presence within these regions, also within the corpus callosum white matter as well. We also assessed the lateral ventricle choroid plexus, and we're seeing just this very distributed profile of enriched T cells in our microbially experienced mice. So then we appreciated, of course, these cells are in the brain, and we wanted to know if they act in kind of a solitary fashion or if they are dynamically interacting with other brain cell types. And of course, I think based on those UMAP projections, the most obvious sort of candidate immune cell to look for interactions would be myeloid cells like microglia or monocytes or DCs. And so we had several ways to approach this immunohistochemistry, flow cytometric data. But for the purposes of this presentation, I wanted to highlight a technique that is quite unique to our lab, which is performing intravital microscopy. And so we decided that we were just going to focus on one sort of set of interactions, CD8s with myeloid cells. And we wanted a technique that was tried and true in our lab that has good signal to noise ratio where we could discern these interactions. And so what we did is we adoptively transferred mice with EGFP expressing Thi1.1 OT1 T cells that are specific for ovapeptide. We adoptively transferred them into reporter mice, including CX3CR1 TD tomato mice and CD11C EYFP mice. We then performed a dendritic cell dendritic cells that were pulsed with ova prime, followed 7 days later with that sort of fourth infection in the specific pathogen experience lineup, recombinant or Listeria monocytogenes that's attenuated, and in this case is going to also express ova, and we boosted those mice. Then at a memory time point, we performed intravital microscopy to determine the quality of these interactions in the brain. So to orient you, what you're going to be seeing here in red is the vasculature, CX3CR1 positive myeloid cells in purple, and then our OT1 T cells in green. And so hopefully this video is showing up okay. And what you can probably first see is we have a lot of circulating memory OT1s in the vasculature that are just sort of very quickly moving by, never sort of sticking around in the brain. But what we do see are these cells that are not stagnant, they are very modal. OT1s that are outside of the vasculature that are dynamically interacting with these CX3CR1 positive myeloid cells. To show just one more video, we also performed these assays in CD11C YFP reporter mice. And here, these cells are comparatively rarer, as we may expect in this tissue. And what we can similarly see is that these cells are dynamically interacting with these cell types just at homeostasis, no antigen around. Okay, so summary so far, in thinking about how to profile these memory T cells in microbially experienced mice, we've to varying extents between these two model systems, looked at number, their functionality, their residence capacity, and briefly touched on some myeloid cell interactions. But I think the bigger question here is what impact could there be of these microbially induced T cells on neurological disease? So stepping back a little bit and thinking about the roles that brain TRM have, I think TCR-dependent roles are a bit more well-defined. We know that if you are able to get pathogen-specific TRM in the brain prior to an infection, that they can durably protect mice against otherwise lethal intracranial infections. And so I think there is a biological motivation for these cells to go in the brain after a peripheral infection or vaccination, such that if a pathogen were to change in tropism or escape peripheral immunity, you have a first line of defense. What we also know is that for certain specificities, if TRM have T cell receptors that are, say, specific to neuropeptide-containing neurons or myelin, that these cells can wield neuroautoimmune disease. But I think an area that is still emerging is sort of what are the TCR-independent roles of brain TRM? In the example that Tomomi was able to present earlier, sort of what's happening in the context of brain homeostasis. These cells may not be seeing their antigen, but they are nonetheless influencing feeding behavior. One way that we wanted to think about this is how could these cells influence brain disease? Thinking about the fact that In the future, I will be in a clinic, and it's very difficult to disentangle or extract what a patient's prior infectious history has been unless they've been hospitalized, and how that could potentially influence their susceptibility or manifestation of a neurological disease state. And so I think there are a lot of model systems that really do warrant the utilization of microbe experienced mice. We considered models of neurodegeneration, aging, stroke, pain, sleep disorders, traumatic brain injury, and seizures. I would say we ruled out some of the first three since we were using young adult mice, and many of these conditions start to manifest in older age. But we did ultimately have to pick one and understand how microbe-induced memory T cells could influence these disease states. We chose seizures, and we were able to collaborate with another MSTP student, Ben Kreitlow, at Iowa to perform these studies. And so the big question here is sort of twofold. How do seizures, when they happen in the brain, influence now these enriched T cells that are sort of witnesses to whatever happens in this tissue? In parallel, we wanted to ask the question of, does being a microbe experienced host and having these T cells actually influence the manifestation of a seizure in the first place? And so first, we're going to tackle this first arrow. So in order to model a seizure, we needed something inducible. And so what we did is we took our specific pathogen-free and experienced mice, and at our memory time point, we induced a 30 milliamp current induced seizure via transauricular current delivered via ear clips. And 2 days later, we wanted to assess, were there any changes in our T cell populations of interest? When we first looked at cell numbers, we did not see any differences following the induction of a seizure. However, we did start to see some differences in functionality. So what we did is we looked at our antigen-experienced CD8s and CD4s with and without a seizure. And what I hope you can appreciate is that there is sort of a pushing of a ceiling effect in terms of the ability of these cells to produce cytokine, where we're seeing an increased frequency of interferon gamma and TNF producing IV negative CD8s and CD4s. We also assessed functionality in a few different ways. We looked at the capacity of these cells to degranulate following stimulation. There was a modest increase after a seizure. Directly ex vivo, so no stimulation condition, we saw that following a seizure, there was a reduced proliferative capacity and reduced mitochondrial mass assessed by Ki67 and mitotracker green staining respectively, suggesting that these neurological events may be influencing the cell health or composition of these cells. And although I don't have time to review it, we importantly saw that if we looked at splenic circulating memory T cells in the same hosts, we didn't see any alterations in numbers, phenotype, or functionality. So then we wanted to ask the reciprocal question, does sort of having these brain-enriched T cells or being a microbe experienced host in general influence your susceptibility or manifestation of a neurological disease? And so to do this, we needed an inducible system to sort of deplete T cells. We couldn't start with knockout mice because they were probably not going to handle this infection regimen very well. And so what we did is we utilized some antibody either depletion or blocking methods to either deplete CD4s and or CD8s, or to block these canonical type 1 cytokine pathways. And then we induced a seizure at a memory time point. I think the most important piece of data in this following graph is the fact that if we compare SPF and specific pathogen experienced mice, we do see a significant increase in overall seizure duration. We see that if we deplete specific pathogen-free mice of their T cell compartment, that they are going to be commensurate with SPF mice, no difference. But what we see if we look at specific pathogen experienced mice that have T cells depleted, they are now going to be restored to sort of normal seizure duration levels. We were, of course, asked to sort of consider individual contributions of CD4s and CD8s. Here we see significant to trending results with single depletion. There's a lot of crosstalk in terms of cytokines between these cell types and influencing the residency phenotypes of each other. So I don't think we can necessarily pin down exactly who's doing what. But at least when we look at our cytokine depletion or cytokine blockade, we see that maybe TNF may be playing a role in this propensity for a seizure to manifest differently in specific pathogen experienced mice. We also wanted to assess whether certain TRM-based signaling pathways could influence the quality or manifestation of a neurological disease like a seizure. And so here we utilize CXCR6 knockout mice in our specific pathogen experience regimen. And we saw that this was a pathway that perhaps did matter in influencing seizure duration, potentially with influencing the sort of CXCR6, CXCL16 axis between myeloid and T cells. And so overall, I think this work is perhaps best summarized by Alexis Johnson and John Lukens in this Nature Immunology News and Views. But to briefly summarize, we've shown that polymicrobial exposure can induce massive numeric increases in brain surveilling memory T cells, that many of these T cells are TRM that interact with myeloid cells at homeostasis, that they are in the brain. And by virtue of being in the brain, they may be differentially impacted by neurological diseases like a seizure that could influence the caliber of these cells. And finally, that microbial exposure and/or T cell signaling pathways can modify the intensity of a later neurological insult. And I think the bigger picture idea here is that studies of basic neuroscience almost uniformly use SPF mice. And in many cases, that may be totally fine to make translation to microbe-experienced humans. But in some cases, it may not. And so something that we're proposing is that perhaps the utilization of microbe-experienced mice may help unveil some of these blockades and trying to understand neuroimmunological phenomena that is occurring in microbe-experienced humans. And we propose that perhaps microbial exposure is a biologically relevant variable that could offer greater insight into these neuroimmunological phenomena. With that, I would like to thank members of the Hardy Lab, Beta Lab, Griffith Lab, Buchanan Lab, the